Welcome to today's USP Pharmatech webinar, Quality Considerations for Aseptic Processing. Thank you for taking the time out of your busy schedule to join us. We have two presenters for today's webinar. Presenting first today is Mike Porter, Vice President, Regulatory Compliance, Pharmatech Associates. Presenting second today is Mark Mitchell, Principal Process Engineer for Pharmatech Associates. Welcome and thank you to both of you. And now I will hand the presentation over to Mr. Mike Porter.
Thank you. And Mark and I both want to thank USP for the opportunity to talk about aseptic processing and some of the quality considerations related to that space. And really, we've divided up the presentation into four key areas. And first, we're going to talk about some of the emerging regulations and changes that are happening. Talk about some of the ongoing challenges related to aseptic processing. Also look at some of the emerging trends and best practices that we see in this space, and then finish with the Q&A portion of the presentation. So to begin, we want to highlight the fact that there is a large body of knowledge that exists around aseptic processing, and it's not just the regulatory guidance that exists. There's also industry standards and industry guides, which collectively inform us around the expected best practices in this space. I think the important thing to recognize is that these documents across the board continue to evolve and change over time. And a really good example of that is the recent changes to the EU Annex 1 from August of this year that will start going into effect in August of next year. But also the industry standards and industry guides are continued to be looked at and refined and cover a large number of very specific topics, whether that be ISO standards or USP standards that exist out there. And these are continued to look at and to revise over time. Also, the industry guides are continue to pull in experts to relook at the information and to update as needed. And I've just listed two examples here, the baseline guide, volume 3 on sterile product manufacturing, showing it's in its third edition, and also the PDA technical report number 13 that was revised in 2022 regarding the environmental monitoring program. So it's incumbent on all of us as we work in this space to maintain our knowledge across this evolving information set that informs us around current best practices in aseptic processing space. And really there's three reasons these regulations evolve that we'll take a look at. And the first is that when we observe noncompliance or risk to patients, it's important to provide additional detail and clarification in these guidance documents and standards so that all can benefit from the knowledge gained from that one situation or that evolving risk that's been identified. And this really isn't significantly different from building codes or fire codes or earthquake codes or other similar types of codes where as we learn, it's important to share and help others receive the benefit of that learning. These documents also evolve based upon having new products come into the marketplace and understanding the special considerations that are related to them, much like cell and gene therapy products, or from technology evolving and emerging like single-use systems or RABs or previously isolators, and understanding how that they uniquely work in this space and how we need to interact with them in a way to really produce the positive outcome for patients that we're looking for. So as these changes occur, we really want to embrace these best practices. We want to share these best practices and really make sure that we have a way to ensure control of contamination in aseptic processing in order to produce a reliable supply of high quality product for patients. As I mentioned, the EU Annex 1 changes kind of crystallized some of this evolution of thinking. And Mark, I know you've been spending some time digging into the changes that have been submitted in this recent version, and I'll let you talk about some of the highlights that are important.
Yeah, thank you, Mike. Unfortunately, the revisions to the EU Annex 1, which is the part of the European good manufacturing practice, specifically referring to the production of sterile products. Unfortunately, the revisions are so extensive, we don't have time to be able to dig into all of them. And maybe in the future, we may have the opportunity to do a separate educational presentation specifically on these changes. As Mike mentioned, the revision was made public in about 2 months ago at near the end of August. Just to give you the scope of just how many changes have been made, the version previously in force was the 2007 version, and it was 16 pages long. The new version in 2022 is 52 pages long and then has I believe, a 6-page glossary added onto it. So all I can do for you today is kind of touch on some of the high-level highlights in the changes. The first one I wanted to talk about is this introduction of what's called a contamination control strategy. And really what that is that it's the desire of EU regulators to have companies put together a document or series of documents or indexed or something, essentially a common point, which will either describe or point to all the appropriate documents that are relevant to contamination control, whether they be risk assessments, facilities, monitoring locations, gowning, sterilization methodologies. Frequently in companies, these have been distributed across multiple responsibilities. And so having a common location to be able to reference and clearly state, what is the strategy that is being used for contamination control, particularly for aseptic products. One of the good clarifications that was made in the recent revision is the clarification between the classification of a clean room, the qualification of a clean room, and the ongoing monitoring of a clean room. Classification is referring primarily to the ISO standard, which is used to determine for EU, it's the grade level of the particular clean room. In the US, it uses the ISO class number. So that's specific to that ISO standard. Qualification is a more general term that not only includes the classification, but also such things as the HEPA integrity testing, the pressure differential measurements, the air changes, the phase velocity, et cetera, that is used to essentially qualify the clean room, and in fact is used now within the annex to re-qualify the clean room on a periodic basis. And then finally, monitoring more has to do with the day-to-day or batch-to-batch assessment of particulates, microbial, process conditions such as temperature, humidity, differential pressures, et cetera. There have been some minor revisions to the particle and microbial limit tables. Most of the revisions in the particulate tables have been in the 5-micron range. There really haven't been any revisions to the 0.5 micron range, mostly to align with different strategies relative to classification of these grade A and grade B for 5 micron versus how monitoring is performed. And then finally, there's a minor revision to the microbial table. Some people might consider it major, but essentially it's referring to the grade A, both in static and dynamic conditions, as having a limit of no growth. Previously, it said less than 1 CFU, which of course less than 1 is 0, but there was some misinterpretation that 1 was acceptable. They're essentially making it very clear that it is a no growth limit. There is 0 is the limit. Somewhat controversial is the introduction of the requirement for PUPSIT. That stands for pre-use, post-sterilization integrity tests. And this has been fairly uncommon in the industry. Usually people perform the pre-use test pre-sterilization, either going with manufacturing documents or their own internal procedures. There is this particular risk that's considered to be performing this post-sterilization testing, potentially risking the sterilization of that filter. There is some allowance within the guidance that a risk assessment can be performed to justify not performing the PUFSIT test. Also somewhat controversial and actually requiring a 2-year implementation, whereas the other sections require a 1-year implementation, is new requirements for how lyophilizers are loaded and unloaded. The requirements are actually pushing more toward an automated methodology for loading lyophilizers and not so much the standard HEPA kind of transfer that is typically used throughout industry. Finally, there's been a requirement added for container closure integrity. There, in fact, has always been a requirement in previous version for 100% testing for those containers that are closed by heat fusion. Now, what is clarified is there's an expectation for any other closure mechanism, such as stopping of a vial, that there be a statistically based sampling plan to determine the container closure integrity of that lot. And again, I'll point out that, in fact, the implementation time frame is rather short. I mean, in fact, it's 2 months have already gone by, and most of the sections are to be implemented by next August. And with the exception of the lyophilizer section, which has got a 2-year implementation.
And so, Mark, even with the broad body of knowledge that exists and the fact that it's revised and kept current, it seems like there still are some aseptic myths and misconceptions that tend to surface on occasion. And we wanted to address a few of those today. So I'll introduce them and have you respond to these. But the first myth or misconception would be that sterility testing is how we prove an aseptic product is sterile.
Yeah, this is a misconception usually for those who are new to aseptic processing. This is a false statement. The sterility test itself is a test of samples from the lot, and only those test samples are actually being proved to be sterile. That sampling plan that is described, particularly in the USP, is not a statistically based sampling plan anyway, and there's really no way to infer that the rest of the lot is sterile simply from the fact those samples are sterile. So exactly. So why do we do that? I mean, we do these samples primarily as a final confirmation that all the other controls that we're doing to assure an aseptic process and a sterile product are in fact in place. This means that actually the failure of the sterility test is more relevant in telling us something than actually the passing.
And I think along these same lines, the next one is a sterility test can prove a lot with a non-conforming event is acceptable.
Yeah, this happens from time to time, usually in people performing an investigation relevant to a exceeding a particle limit or a microbial limit in a critical area, that they rely too exclusively on the sterility test. As we explained previously, the sterility test in itself only tells us that that sample is sterile and really doesn't give us any more information about the rest of the lot. The appropriate response to these kind of non-conforming events is really to perform a complete risk assessment on all the different series of controls that you have in place. Those are much more relevant in making the assessment of whether the lot is acceptable, as opposed to simply just looking at the sterility test.
Very good. And let's talk a little bit about media fills and the myth and misconception that a media fill can be performed prior to qualification of other processes.
Again, this is false. This is, again, the tendency of people of focusing on things like sterility tests and media fill in terms of aseptic processing. Really, the media fill is a simulation of what is supposed to be your controlled process that you normally perform every day. If it is supposed to be the normal process, then it has to be the normal process. It has to be the qualified equipment. It has to have all the sterilization in place. It has to have the facility qualification in place. It has to have the trained personnel in place. That media fill is only telling you something useful if, in fact, all those other controls are in place. If those controls are not in place and you pass the media fill, that passing really doesn't tell you much of anything.
And continuing with media fills, another myth is a media fill can prove equipment or procedures are suitably aseptic.
This again is false. Really, the purpose of the media fill is to qualify particular operators on particular equipment performing particular interventions. Um, Equipment and procedures must be specifically designed for aseptic use. You can't just put any piece of equipment and run it and run a media fill and say that you pass. There is an expectation that equipment must be designed with the proper airflows, with the proper surface finishes, with the proper interactions with operators, materials of construction, etc. The same thing in regards to procedures. I mean, really only aseptic technique that has very specific requirements in how it's performed are suitable for the aseptic environment. You can't just simply put a non-aseptic technique within a media fill, have that media fill potentially pass by random luck, and then claim that somehow that procedure is allowable in a media fill.
And finally, let's talk about the myth that a total particulate count can predict a potential microbial contamination.
This is a tricky one. This is actually partially true. What I'm talking about is a total particulate count. Previously, we used to call these non-viable particle counters. The reality is they're counting all the particles, whether they're viable or they're non-viable. The reality is a microbial contamination is related to particulates. They're associated with particulates. It doesn't mean all particles are microbial contamination, obviously. The issue of why we focus on total particle counts is that we can do total particle counts in almost real time. We can have online monitoring, we can have online alarms, we can tell the operators immediately whether you've exceeded these particle counts, and then you can respond. And there is a general correlation that a higher level of particle counts is a greater risk of microbial contamination. It's not a perfect correlation, but the reason we depend on that is that we can't tell whether the microbial contamination is there until we see the microbial results, which unfortunately are several days later. They have to be collected and incubated on media in order to be able to be detected. So that's why we depend on those total particle counts. That's why we evaluate those total particle counts in real time. And we're trying to predict in advance the potential risk of microbial contamination. Just in general, to kind of underline what we were stating before with these myths and misconceptions, is that really I like to look at all of aseptic processing as this kind of pyramid. I mean, we don't rely solely on that sterility test. We don't rely solely on that media fill. Over focusing on those upward elements is essentially ignoring the fact that they really only have value because there's underlying controls in place. And that's kind of what this pyramid is showing. There's a whole series of systems, controls, processes that have to be in place in order for us to rely on sterility tests and media fills. Fundamentally, at the bottom, we have to have a pharmaceutical quality management system. We have to have documentation control. We have to have training. We have to have personnel qualifications. We have to have proper facility designs. We have to have those underlying systems that we can now build on. On top of that, we have we design our facilities to particular standards, our equipment. We train our personnel in gowning and aseptic technique. Even though we perform all these things according to standard, we still must perform the classification of the environment. We have to qualify the facility. We have to qualify the personnel. We have to qualify the equipment. Then once we've done all the qualification, we have to maintain it. We have to maintain that microbial control. We do through cleaning, disinfecting our facilities, our equipment, and then on top of that, now we have to start be concerning about how do we monitor? How do we know we're being effective ongoing with our cleaning and disinfecting? We have to do particle monitoring. We have to do a process monitoring, and we have to do that on an ongoing basis. On top of that, of course, now we rely on sterilization methodologies, whether it be gamma, steam, autoclave, ETO, whatever, and the very important for most products, the sterile filtration. Once all those foundational elements are in place, now we can do a media fill simulation. And once we have a media fill simulation to show that we have a process that we can control, then we can rely on that sterility test to tell us that all these elements are in fact giving us our aseptic process.
So, Mark, with the broad body of knowledge that exists and this expectation of a strong foundational system in place to support aseptic processing, we did want to highlight a few of the findings that are commonly seen and then talk about some of the emerging trends that may be addressing those. So, in case of facilities in particular, there are a number of older clean rooms that have not been reclassified since the initial construction, or some of the aging HEPA filters have not been proactively replaced. Adding equipment without regard to room airflow, so continuing to make changes and evolve the space without consideration of some of the critical elements related to aseptic processing. And also the lack of pressure monitoring between the clean room and unclassified areas like emergency exits. So these in particular may reflect back more on some of the older facilities. But Mark, what are some of the trends that we're seeing to kind of help with our facility issues?
Yeah, we consider these trends to be kind of some of the best practices that are kind of out there that should be considered to be embraced. First is the continuous monitoring of room conditions, such as differential pressure between rooms of different classifications, with a visual and audible alarm for the operators. We're essentially talking about a horn or a light within the room. This is something so that if any issue occurs in regards to the facility operation, that the operators have an immediate notification so they know that they can either stop the process or protect the product in some way. Also, we're starting to see this more complex, what's called CFD, or computational flow dynamics, to assess airflows in clean rooms. This is more in use of new clean room designs, and using this computational model to not only determine the ideal location of HEPA filters and returns, but also when we introduce equipment and also personnel. And these systems actually have models where you can insert a person and a typical particle generation considered for a person, and you can actually see where the particles are actually flowing within the clean room and where the problem areas potentially can be and how we can deal with that. Also, this is primarily in regards to some of the revision that was done to the EU Annex 1, which is the periodic reclassification for the HEPAs. Now, typically, the reclassification had always been done for things like HEPA integrity and differential flow and differential pressure flow and air changes and things like that. But now that Annex 1 revision is actually saying requalification includes reclassification according to the ISO. And that effectively means the ISO requires you to grid the room out in a particular way and bring portable particle counters in and make those very specific measurements to assure that those spaces still meet the ISO requirement or the specific requirements that are in the EU Annex 1.
And as we look at equipment, some of the repeating trends or findings that we might see are the conversion of a filling machine to an open RABS without regards to airflow visualization, or some of the operator training related to the use of the glove ports for interventions, as well as the failure to really provide local protection air for uncrimped lyophilized vials.
Yeah, the ongoing trends in regards to equipment. We're of course seeing greater use of isolators or other barrier systems like RABs. In fact, if you are installing a new filling line or lyophilizer or such, essentially you're going to be asked by the regulators is why are you not using some sort of barrier system, a RABs or an isolator. Also, we're seeing more and more automated lyophilizer loading, where personnel do not have to manage the vials or put them together in trays and then transfer them with HEPA carts into the lyophilizer. Instead, it's an all completely integrated system going from an isolator all the way directly into the lyophilizer, loading and unloading without direct intervention of personnel. Also, we're seeing, particularly on new filling lines, more use of these pre-washed and pre-sterilized vials. They're either provided in trays or tubs that are pre-wrapped. And the intent here is to essentially bring these, pre-washed and sterilized systems into isolator environments so that you don't have to install things like the vial washer or the depyrogenation tunnels. And finally, within biologic processes in particular, we're seeing greater use of the pre-sterilized single-use systems with these intrinsic aseptic connectors that don't require a critical area in order to perform an aseptic connection.
In regards to microbial monitoring, we know that regulators are very much focused around risk assessments and robust scientific justifications. And I think this is one of the findings that we see in this space around the lack of a solid justification for the sampling locations. Inspectors are also looking at historical results and the ability to trend those and use those effectively in the process. And again, here the finding would be the failure to use those trends when setting alert limits based on historical data. There's also concern for the use of active air samplers in the critical zone without consideration of sampler body disinfection and exhaust HEPA filtration.
Some of the trends we're observing is we're seeing a lot of the settling plates, particularly in critical areas, the settling plates being removed with quite frankly, a much more effective mechanism for collecting a microbial air sample, which is the active air sampler, which uses a vacuum system. The settling plates are very, very limited, particularly in a critical area. In fact, they've been assessed by the FDA and the EU as primarily a qualitative assessment and not a quantitative assessment of microbial measurements of an area. And in regards to these active air samplers being used in the critical zone and their disinfection of the body, we're seeing more and more people use these remote systems where essentially either the active air sampler media is in a disposable or reusable stainless steel piece, but the actual vacuum air pump that actually pulls the air through onto the media is in fact located remotely and outside of the critical zone. So it eliminates the need of the sampler body disinfectant.
And for total particulates, the failure to review the total particulates during release continues to be a finding, as well as not monitoring particles during aseptic assembly. And again, we see similar comments and findings around the justification of monitoring locations, as well as the trending or setting alert limits based on historical trends. And then finally, undetected failure of remote particle monitors is also another item on the list.
The trends, of course, now is to no longer use these portable particle monitorings, but actually have continuous particle monitoring with permanently mounted locations. Again, when we talked about that myth in regards to total particles, we want an immediate visual and audible alarm for operators if in fact the particle limits are exceeded, so that they can respond immediately, whether that is to stop an equipment, stop an intervention, or segregate product. Particle monitoring locations, they really need to be done by some sort of risk assessment regarding the room in terms of the flow, the activities, the location. Again, we talked that we have this computerized tool, particularly for new clean rooms, of CFD, computational flow dynamics, which can help us identify in a model where the problem areas are and where the best locations for locating particle monitoring and even microbial monitoring. Finally, in the past, what's happened is that the particle monitors that were being added to a facility were in fact being integrated with things like BMS systems that were not specifically designed for particle monitoring, particularly in how it dealt with the data and actually triggered the alarms and in fact generated batch reports. So we're seeing really the trend of using this specifically designed particle monitoring system that has very specific software with very good batch reporting so that you get a very easy to read and easy to understand report on lot release so that the quality professionals can assess that in fact the particle limits had not been exceeded.
And Mark, our last area is around disinfection, and of particular concern here, I think the failure to qualify disinfection efficacy is a finding that shows up in quite frequently, as well as the over-reliance of IPA as a disinfection agent, and the lack of using multiple disinfection agents with differing modes and as part of the disinfection process. Properly cleaning disinfection residuals is identified, as well as aggressively responding to detected mold and people not taking the appropriate steps when there is a positive detection. So, Mark, what would be some of the trends in this space?
We're seeing more and more use of these pre-sterilized and pre-mixed disinfectant solutions. What happens is they're either in a system that can be mixed or already are mixed in advance, and then they're already pre-sterilized, usually by gamma. They're triple bagged so that they can be easily brought into a clean room. As opposed to what previously happened, people would have to mix their own disinfectant solution. There's potentially an error that could happen. And then they would have to sterile filter each one and do the filter integrity test and to assure that you basically had sterilized disinfectant solutions. Also because of, I think, primarily COVID-19 kind of introduced this thing of this hydrogen peroxide misting or spray systems to be able to cover large areas like floors and walls and ceilings and stuff with a disinfectant solution of hydrogen peroxide.
And we'll transition now, Mark, to talking a little bit about some of the technology and the advantages and challenges of those technologies in the aseptic processing space.
Isolator technology is not necessarily new, but I think it's gotten better and better. You have more vendors to choose from, and their expertise in being able to build those isolators is much better now. Of course, when we talk about something like an isolator or barrier system, we're trying to isolate the operator's environment from that aseptic equipment, that aseptic process. And this is particularly important, not just from an aseptic standpoint, but also when we talk about potent compounds. Consequently, if we're doing that isolation aseptically from the environment and the personnel, that means the surrounding room classification can actually be a lower level. In fact, for the FDA, the surrounding environment switches from a ISO 7 down to an ISO 8 if you are using an isolator. Consequently, if the classification is dropping, also the gowning requirements are less. Also, the airflow and air changes requirements are less. So there's an expense of the isolator, but there can be some savings regarding the room surrounding it. Also, isolators use this very well-controlled sterilization process. A lot of studies have been done in showing that the effectiveness of this hydrogen peroxide vapor sterilization methodology much more well controlled than a simple disinfectant type wipe down process. Some of the challenges we face with isolators still, these are nice, well controlled hydrogen peroxide vapor processes to do this disinfectant, but the cycle times, depending on the equipment, can be rather time consuming. You have to do dehumidification, then you can perform the sterilization, and then you have to exhaust those vapors. That can be quite extensive, and of course, that sterilization time is time that cannot be used for production. Because isolators are essentially closed systems, in order to be able to get materials into them and out of them, we have to use very well-defined entry and exit methodologies, whether they be rapid transfer ports, whether they be airflow entries, whether they be shuttles, whatever. But we have to get those unfilled containers into the isolator, and then we have to get those filled containers out of the isolator, potentially to even a lyophilizer. Integrating with lyophilizers are very much, open and closed kind of door kind of situations. They have longer cycles, sometimes in the filling. So integrating filling lines, sometimes multiple filling lines to multiple lyophilizers when you're using an isolator system can be rather complex. Also understanding that when you do an isolator and you seal that isolator and you perform that hydrogen peroxide process, that process is now sealed. You still have glove ports, you can still do interventions, but in fact, you're very limited in what interventions or maintenance or anything like that you can do during that process. So you have to be assured that a process that's within an isolator, that equipment must be fully developed, it must be very reliable. If it's not reliable, an isolator process is really going to be very frustrating. We also see more and more use of single-use systems. They were really very heavily embraced in biologics, but now we're seeing them more and more, particularly for sterile filtration into a bag and then connecting that bag up to an isolator or RAB system. So these closed systems, again, they're isolated from the operators in the room. Because we're using these disposable bags, we don't have stainless steel tanks that require a CIP and SIP, which we have to develop and validate. And then what is particularly some of the newer technology are these intrinsically aseptic connectors. These essentially allows us to connect different single-use systems together without having to use a critical zone. So we can actually perform aseptic connections without having to have a grade A or ISO 5 zone. Some of the challenges with single-use systems, you're really turning over that cleaning and sterilization to a outside vendor, a supplier. And so you're relying on their processes, which you really should very rigorously audit. Particularly the sterilization processes. If you're using gamma, there is a requirement of doing quarterly sterilization dose audits for those systems. It's not always clear that when you buy these single-use systems that in fact is being done. So that is a bit of a challenge. These are polymeric systems. They're polymeric bags. They're much better than what they used to be. They usually are pretty compatible with most products, but you still need to understand what the extractable and leachable profile for your product is going to be in that system. There's always a potential risk of loss of integrity. These bags are fairly robust and they do have containment systems to help with this, but they're polymeric systems. There can be manufacturing defects. There potentially is a risk of a puncture. There are integrity testing systems for bags, but they are very cumbersome and complicated to use. There is always a potential risk of adding particulates to the product because these are polymeric bags. This risk is probably not as significant as it had been in the past. Then, of course, of using a single-use system, it's a single-use system, so you use it once, and then you have to take that bag and you potentially have to dispose of it, potentially as hazardous waste. So it can be rather costly. In terms of cell and gene therapy, I mean, I'm not an expert in the area of cell and gene therapy. So in regards to the advantages and issues regarding the process of cell and gene therapy themselves, I would actually reference some of our co-workers have actually put together another presentation with USB education regarding cell and gene therapy. In fact, they just presented it last month. But in particular, what we can talk about is some of the issues and challenges with aseptic processing in regards to cell and gene therapy. We know that we can't take cells and we can't sterilize them. We can't do terminal sterilization like heat or radiation. It's just going to destroy the cell. So we know that that's just not an option to us. So aseptic processing is what we have to do. Most aseptic processing with small molecules are in fact done with sterile filtration. Unfortunately, large molecules in cells, they're just too big. They're not going to work with sterile filtration because you'll actually remove your actual cells. There are wide use of using these single-use systems. Not only using these systems, but doing these aseptic, intrinsically aseptic type connectors without using critical zones. Media fills are a bit more complicated. Media fills were kind of designed around the fact of filling lots of vials or syringes or ampules or whatever. So you'd have thousands and thousands of containers to be able to assess, potentially a low level of contamination. In cell and gene therapy, of course, when you do a media fill, usually you're ending up with very few containers, usually only a handful. So being able to assess in the same way low levels of contamination is a bit more difficult. Some final thoughts. As we understand, as our regulations shift, but really aseptic processing is always bringing on these new technologies, these single-use systems, more isolator systems, new products and new technologies. They're always happening and always being introduced. So luckily, we have all these different approaches and abilities to be able to do contamination control, as our diagram kind of shows. So it's our responsibility to stay vigilant and to learn about these changes of technologies, the changes of products, the new approaches to doing contamination control and aseptic processing in order to be able to use all these items effectively.
Yeah, Mark and I would agree completely with this thought of staying vigilant. We do have a large body of knowledge. It is continuing to evolve. We need to have strong foundational systems and processes in place, as you showed in your pyramid, and these can continue to change as well. So staying abreast of these changes and on top of the emerging trends continues to be important in producing this reliable supply of high quality product for the patients at the end of the day. And hopefully pulling these pieces together today will help as you continue on that journey. Mark and I have provided our contact information at the end in case you would like to reach out after, but we will take a few minutes now to address the questions that have been put into the tool while we've been presenting. And so, Mark, it looks like the top question here is, what are the sterility requirements to assess preclinical studies for gene therapies made with cell culture?
Yeah, I mean, one of the issues I need to point out is what specifically is meant by a preclinical study. If it in fact is a human study, that it has to be a sterile product. And if you cannot terminally sterilize, then you have to do aseptic processing. So as we can show, that aseptic processing is rather substantial. It's not just simply performing media fills or doing sterility tests. It means having all these other controls in place so that we can rely on those media fills. And so really the phase of what you're in, whether you're in preclinical or clinical or commercial, is not really relevant. What is the issue is safety to the patient. And that's really what aseptic is about.
For infusions filled using BFS and FFS technologies, is there a need to do terminal sterilization at the end? And are there any references?
BFF, blow fill seal, and FFS, form fill seal, just to be clear. When we talk about terminal sterilization, the expectation is if you can perform terminal sterilization, that is what you should be doing, whether it be by heat or gamma or ETO or whatever. That is the approach that has to be done. You get a much higher sterility assurance level with terminal sterilization than you can with aseptic processing, right? Now, prior to terminal sterilization, we have to understand the control of our bioburden. leading into that terminal sterilization so that we know we're doing our terminal sterilization effectively. And then frequently, sometimes I find in processes that the easiest way to assure a consistent controlled bioburden is to perform pretty much an aseptic process. Obviously, an aseptic process is designed to give us the lowest level of bioburden.
Okay. And in regards to sampling, how many samples are required for cell and gene therapy product sterility? Is it the same as USP 71 Table 3?
Certainly, you have to start out with USB 71 to determine the amount of samples and the number of samples. Understandably, with cell and gene therapy, the batch sizes are relatively small, and sometimes the sterility requirements may consume the entire quantity of the batch, which of course is not acceptable. You really have to have a discussion with your regulator in terms of potentially doing fewer samples. As we kind of pointed out, the sterility test is really just a final confirmation of all the other controls. It's not the fundamental methodology of determining whether something is aseptic or sterile.
Okay. And what are the minimum considerations for raw materials to enter a clean room for phase 1 processing?
Again, it doesn't matter what phase we're at, whether phase 1 or whatever. We want to do contamination control if we're doing aseptic processing. Um, for raw materials to be used in an aseptic process, if we're talking about the containers and the container closures, there is a requirement that they have to be sterilized. Right? It doesn't matter whether it's phase 1 or whatever. If it's for human use, has to be aseptic. Yeah, you have to have sterile containers. Other raw materials, yeah, again, they have to be sterile if they can't be introduced prior to the sterile filtration.
And Mark, I think it is a key point to emphasize that human use is human use, and it's irrespective of whether it's phase 1 or phase 2 or commercial, right? If we're talking about use of products in humans, then the considerations are relevant across the board. So fair point to highlight that in the
Yeah,
scope of the
and
question.
sterility testing alone in itself does not mean much. And so you can't just simply say, well, we tested it sterile so we can use it for humans. That's not an acceptable approach.
That foundation of strong process to control the processing steps is important. Agreed. It can't just be based on the final testing. And the last question here, as we're getting ready to wrap up our time, is: are the guidelines associated with endotoxin reduction in regards to aseptic components processes? Are there guidelines? Any guidelines?
Yes, there is a specific section that, particularly in the FDA guidance, that describes what is required for endotoxin reduction. I believe it's a 3-log by spiking that has to be shown. But that is specifically, there is a specific section in the FDA aseptic guidance in regards to that.
Okay, and with that, we will conclude the presentation today. We thank you for your engagement and for the questions. If the additional questions that were not addressed in this will be provided after the presentation, answers to those. But thank you for your time, and please keep an eye out for future USP education offerings like this.